Hi, hello to all the second year BIPC students. So, here we are to discuss the major important questions from microbiology, included bacteria and virus. So, before that I would like to tell you that from NEET World we have launched all the IPE books which are completely for free of cost, which are available in both online and offline platforms. If you want to download the soft copies of our books, what you have to do is very simple process ma. First you have to go to Telegram and then Instagram, follow our channel, that is NEET World 25, both in Telegram and Instagram. The added on benefits that you get apart from IPE is you will get the creative reels, shorts and more replica questions based on your previous year questions in both the Telegram and Instagram channels. And then from Play Store you just download the NEET World app, and in YouTube NEET World 2025 is our YouTube channel. You just follow us in YouTube as well. In Play Store, just download the NEET World app and say hi in the chat box. You will get the complete PDFs of all the 4 materials for free of cost. Okay, and then if you want to know more about the programs that are being running over here, you can call us to the given number, and to know more about the institute, you can visit our official website. Okay, which is over here. Okay, this is our official website. This is the complete structure which I can give you to extract the books in online. Okay, just follow us in all the social media platforms and install the app, say hi in the chat box, and then you will get the access of all the 4 materials. Now let's get started with the concept. Yeah. First is about microbiology. In that we are going to deal with the chapter bacteria. Here we go with the first question, which is major, important and asked multiple times. If you can see from May 2016, March 2016, March 2018, 3 times asked question. Okay, how bacteria are classified based on the basis of number and distribution of flagella. Okay, how bacteria are classified on the basis of number and distribution of flagella. Here if you see, flagella are whip-like locomotory structures. Okay, they are whiplash-like locomotory structures that help the bacterial movement on the basis of number and arrangement. We have 5 types. Okay, so if at all there is only one flagella that is attaching to the bacteria, it is addressed as what ma? Monotrichous. Okay, if 2 flagella are there on the either ends of the bacteria, then it is addressed as what? Amphitrichous. Okay, if bunch of flagella are there only at one end, is known as Lophotrichous. Okay, if flagella are present completely surrounding the bacterial cell, then it is known as what? Peritrichous. Okay, first is monotrichous, where it has a single flagella attached to the bacteria. Amphitrichous means bacteria has 2 flagella on 2 ends, either ends. Lophotrichous, bunch of flagella at one end. Peritrichous, complete bacterial body surrounded by flagella. Such type is known as what ma? Peritrichous flagella. Now you see the description and examples are important. Just by looking into the diagrams you can easily make out the types, but look into the examples properly. First, atrichous means bacteria without flagella. The best example you can give over here is lactobacillus or lactic acid bacteria. Clear, right ma? Second example you see, that is monotrichous, which is single bacteria. Comma shaped bacteria, this you can see the example, that is Vibrio cholerae, which helps in what? Directional movement, single directional movement, as it has single flagella. Next is Lophotrichous, 2 or more flagella only at one pole or at the one side. The tuft of flagella will work together and this will help the bacterial movement to move in a fast way. Okay, this will adhere to the fastest movement of the bacteria. Best example you can give is Pseudomonas fluorescens. Amphitrichous, single flagellum or tuft of flagellum which is present at both the poles of the cell. This bacteria, they can move in both the directions. Based on its requirement, it can move in both directions. Example you can take is Pyrillum volutans. Next is Peritrichous. Many flagella are distributed all over the surface of the bacteria. These bacteria will show the rapid movement. Okay, Peritrichous, This type of bacteria will show rapid movement in all the directions. If you see the example, Escherichia coli, that is E. coli. Okay. So, here what you have to remember and highlight beta, first is the diagrams, all the 5 types. You can also mention a single flagella without the flagellum, atrichous, monotrichous, amphitrichous, lophotrichous and peritrichous. And directional movement of bacteria, whether it is single direction or 2 directions, or is it surviving with the fastest movement. Okay, these things should be highlighted. And then about examples. The example of atrichous is lactobacillus. Monotrichous is comma-shaped Vibrio cholerae. Lophotrichous is Pseudomonas fluorescens. Amphitrichous is Pyrillum volutens. Peritrichous is Escherichia coli. Till here everything is clear, right ma? Next you see what are the nutritional groups of bacteria based on their source of energy and carbon. Of course, it is asked only one time in your previous paper, if we see, that is May 2015. One thing is more attractive ma, if you can present a flow chart here about nutritional groups based on the source of energy and carbon. First you present a flow chart and then you go on explaining the concept. Flow chart here, if you see, bacteria based on nutrition, it is of 2 types, right? Photoautotrophic and chemoautotrophic. Okay, photoautotrophic and chemoautotrophic. If you take heterotrophs, again of 2 types, heterotrophs are of 3 types. What are they? Symbiotic, parasitic and saprophytic. Fine, right? So, as he asked, based on the source of energy and carbon, we have both photoautotrophous and photoheterotrophous, chemoautotrophic and chemoheterotrophic. Here we go with the explanations. First, bacteria will exhibit great diversity in nutrition. Even in your objective, if there is a question like which kingdom from Whittaker's 5 kingdom system of classification, which kingdom will exhibit vast amount of metabolic diversity, then your answer should be bacteria. Which kingdom is exhibiting maximum amount of nutritional diversity, your answer should be bacteria. Okay. So, bacteria exhibit the nutritional diversity, respiration diversity, okay, and metabolic diversity on whole. Okay. Remember that in the objective perspective as well. So, depending on the source of energy and carbon, bacteria are classified into 4 major nutritional groups. First we will see photoautotrophs. In each nutritional group, you have to highlight what is the source of energy. Is it the sunlight or is it from any organic or inorganic compound? And what is the source of carbon? Is the source of carbon an organic compound or inorganic compound? As such, you have to group and mention it accordingly. Photoautotrophs. Photoautotrophs, here we go. These bacteria contain photosynthetic pigments. This bacteria will contain photosynthetic pigments like bacteriochlorophyll or chlorophyll, and they use what? They use sunlight as their source of energy. Remember, photoautotrophs is using what energy ma? Sunlight as energy. And what are the photosynthetic pigments it has? Bacteriochlorophyll or chlorophyll. Fine, right? Next, they obtain carbon from atmospheric carbon dioxide. Okay, they will obtain carbon from what? Atmospheric carbon dioxide. They carry out photosynthesis similar to that of green plants, but they may not always produce oxygen. So, it can conduct both oxygenic photosynthesis or anoxygenic photosynthesis or anoxygenic photosynthesis. Clear, right? Here we go with examples. Examples you see ma? Chromatium, chlorobium. Chlorobium can be green sulfur bacteria or purple sulfur bacteria. Next is chemoautotrophs. Chemoautotrophs, this bacteria will obtain energy from oxidation of inorganic substances. As it is from the oxidation of inorganic substances, they play major role in mineral cycling as well. So, here we go with the point that is it gets energy from where? From oxidation of inorganic substances like ammonia, nitrate, sulfur and iron compounds. So, those bacteria which exhibit chemoautotrophic nutrition will play major role in mineral cycling as well. Carbon derived from carbon dioxide again, but energy source is different. Here in photoautotrophs, the energy source is sunlight. Here in chemoautotrophs, the energy source is oxidation of inorganic substances. Next, they perform chemosynthesis. They perform what? Chemosynthesis instead of photosynthesis. Okay, instead of photosynthesis, they perform chemosynthesis. Examples: Nitrosomonas, Nitrobacter, Bigiatova and Methanogens. Okay, Nitrosomonas, Nitrobacter, Bigiatova and Methanogens. They help in nitrification and they play vital role in mineral cycling always. Okay. Next is photoheterotrophs. This is third class. These bacteria use sunlight as source of energy. They will use sunlight as source of energy, but they obtain carbon from what? Organic compounds. They obtain carbon from organic compounds like fatty acids and alcohols. Fine, right? They do not release oxygen. Okay, they do not release oxygen during photosynthesis. So, they are what? Anoxygenic. Okay, they are anoxygenic. They do not release oxygen during photosynthesis. Examples here you see for photoheterotrophs: Rhodospirillum and Rhodopseudomonas. These are the examples of photoheterotrophs. Till here everything is clear, ma? Now we see chemoheterotrophs. The bacteria derive both carbon and energy from organic compounds itself. But here photoheterotrophs, photo as the energy is from sunlight, whereas carbon is derived from organic compound. Here chemoheterotrophs, chemo means the energy is from organic compound, carbon source is also from the organic compounds. Organic compounds like glucose, amino acids and fatty acids. Clear, right? Very simple. Photoautotrophs, chemoautotrophs, photoheterotrophs and chemoheterotrophs. Then you see energy is released through respiration or fermentation. Okay, energy is released through respiration or fermentation in the form of ATP. Okay, they are further divided into two types. Okay, they are further divided into two types. We will see that. Saprophytes and parasites. Okay, chemoheterotrophs are further divided into saprophytes and parasites. Saprophytes will grow on dead and decaying organic matter. Best examples for saprophytes you can take is Bacillus. Parasites, if you see, they grow on or within the living organism. They grow on or within the living host and they will be causing what? Diseases. Examples you see here: Xanthomonas, Salmonella and Delovibrio bacteriovorus. Fine, right? Everything is clear here, right? So, see. Photoautotrophs, chemoautotrophs, photoheterotrophs and chemoheterotrophs. Chemoheterotrophs again include what? Saprophytes and parasites. Here we go with next. Explain the process of conjugation in bacteria. Conjugation in bacteria. First I will explain with the diagram and then we will go on with the theoretical explanation. First we will see the importance of this question. Multiple times asked. If you see in May 2024, February, March 2024 and March 2025 as well. First you have to identify what is donor and what is recipient. You have to mention that clearly in your diagram. Donor is F plus strain and recipient will be F minus strain. Okay. F plus strain are those bacteria whose plasmid DNA will have F factor. The plasmid DNA will have F factor. The F factor from F plus strain is transmitted to the plasmid DNA of F minus strain. It is being transferred. Okay. So, here this pilus from donor should be extended. This pilus pili is also called as what? Sex pili. or true pili. This will be adjoining the two strains, both F plus strain and F minus strain. As such, from plasmid DNA, the F factor will be sent to the F minus strain, and at the end you can see the donor is also F plus strain, and recipient after receiving the F factor, this is also having F plus strain. Okay. This is the sequential explanation of what? Conjugation process. After giving out the diagram, I always advise you or insist you to first present the diagram and then go for the explanation. Conjugation type of sexual recombination is important not just for your IPE, it is also important for your MSET and NEET. Here you see my definition. Conjugation is the process of transfer of genetic material. This is the process of transfer of genetic material from one bacterium to another through what? Direct cell to cell contact. Okay. And how is it discovered? It was first discovered by Lederberg and Tatum, okay, in the year 1946. Firstly, it is observed in Escherichia coli. And then F factor, if you see, some bacteria will possess extracircular DNA, that is what? Fertility factor, which is F factor or plasmid. Types of cells. We have two types of cells. Donor cell are F plus cells or F plus strain, we will say. Recipient cells are without F factor. We will address this as what? F minus factor or F minus strain. Sex pilus formation. How a sex pili is formed? F plus cells will produce a sex pilus that attaches to F minus cell to establish first contact. It will first establish the contact, and then conjugation bridge, a cytoplasmic bridge where conjugation tube is formed between F plus strain and F minus strain, that is in between donor and recipient cells. A conjugation tube is formed. Now, transfer of DNA. A single strand of F plasmid DNA will pass from F plus donor to F minus recipient through what? A bridge. That bridge is addressed as what? Conjugation tube. That bridge is addressed as conjugation tube. Now, replication. The transferred strand will replicate in both the cells by rolling the circle formation, and forming complete plasmids. Okay? Complete plasmids are formed as such. Next. Lastly, the recipient F plus cell and F minus both will act as donors. Now, as both of them will get that F plus strain, what is the significance of conjugation? Conjugation will introduce a genetic recombination and a variation which will aid in spread of beneficial traits of antibiotic resistance in bacteria. So, all the points in a sequential pattern you have to explain better. Your theoretical explanation goes easy if you present the diagram first. Okay? Just go through the answer. If you are having any doubt, you can ask me. Now, virus. From virus we have three major important questions. First, if you see, what is ICTV and how viruses are named? Important, quite a times asked in your previous year IPE papers. Like if you see in March 2023 and March 2019. The answer here it goes like this. First, ICTV means what ma? International Committee on Taxonomy of Viruses. Okay, it is International Committee on Taxonomy of Viruses, which is nothing but a global authority which is responsible for classification and nomenclature of viruses. First you have to define the abbreviation, that is ICTV. Next, ICTV will recognize three main hierarchical levels of classification. First is family, then genus, then species. Some families may include subfamilies as well. Okay, some families will include subfamilies as well. For example, family names end with suffix viridae. Okay, family name ends with the suffix viridae. Next, genus names end with the suffix virus. Okay, genus name will end with the suffix virus. Species names are written in common English, describing what? Describing the virus and diseases it causes. So here suffix of both family and genus are important. Now here you see how viruses are often named by the disease they cause, for example poliovirus, and by the host organism that they infect, like TMV, tobacco mosaic plant infection, is caused by tobacco mosaic virus, and the type of nucleic acid or site of infection where they are infecting. For example, here you see family Retroviridae, the suffix you see it is viridae. Lentivirus, okay, here you see the suffix is virus, and species HIV, human immunodeficiency virus. Clear, right? Just highlight the suffixes of both family and genus and give out a standard examples accordingly. Very simple. For short answer type of question, your answer should be accurate and specific. Examiner will not look for your lengthy answers. Your points or the answer should be very accurate, okay, and it should be brief, accurate, in specific to the question. Then only marks will be awarded in your exam, okay. Next, explain the structure of TMV. TMV structure multiple times asked question, multiple times. So here if you see, first explain the structure of TMV asked in 2017, okay, in two different months, May and March 2017 you got this question, okay. So here we go with TMV is what? This is one of the most studied plant viruses, okay. This will infect tobacco and tobacco related plants. It is simple yet well organized structure, okay. TMV is well organized structure. TMV is rod shaped or cylindrical virus, okay. This is rod shaped or cylindrical virus with a rigid helical structure. It has rigid helical structure. It measures about how much nanometers length? 300 nanometers length and 18 nanometers in diameter. Remember, length and diameter is very specific, you have to mention it for sure. And what is its molecular weight, ma? Molecular weight is approximately 39 into 10 to the power of 6 daltons. You have to mention it in specific. TMV possesses hollow central lumen, okay. This is your option, okay. Whether you are mentioning the lumen diameter or not is optional, but these 2 points should be mentioned in your answer. What is the length and diameter of TMV and how much molecular weight is it having? How many daltons molecular weight, okay. Then this is the structure of TMV. Always I advise you to first draw the structure, then go on for the explanation. So TMV has single stranded RNA with complete protein code called capsid. This protein code is called as what, ma? Capsid. Protein code has protein subunits. Each protein subunit is addressed as what? Capsomeres. Here you see capsid is a protein code. The virus is surrounded, virus is completely surrounded by a protein code called as what? Capsid over here, okay. This is known as capsid. The capsid is made up of 2130 identical proteins. Remember, very important. These protein subunits are known as what? Capsomeres. Fine, right. These capsomeres are arranged helically. What is the arrangement of capsomeres? Helical arrangement around the central RNA core. Protein subunit, you see, each protein subunit, okay, protein subunit is capsomere here. Okay, each capsomere is composed of 158 amino acids, okay, forming out a protective sheet. Genetic material, if you see, it has single stranded RNA. RNA is spirally coiled, which is inside the capsid. It has 6500 nucleotides, okay. It has 6500 nucleotides, and RNA carries genetic information necessary for viral replication. Here an add-on point for your NEET and MCET key, they may ask you the ratio of number of nucleotides and protein subunits that are present in a TMB. What will be the ratio? Okay, ratio of number of nucleotides is to number of protein subunits, that is capsomeres present in a TMB. Okay, nucleotides are 6500 or 400 nucleotides approximately. Protein subunits are 2000, 130 or 140, which is approximately 3:1 ratio. Okay, the ratio of number of nucleotides in single stranded RNA and number of capsomeres in the capsid of TMB will be 3:1. Okay, what is the ratio, ma? It is 3:1. Clear till here? Any doubt anywhere? Yeah. Next you see function of components. What is the function of components? The capsid protects the RNA from environmental damage. Yes, the RNA encodes proteins which are essential for infection and multiplication within the host cells. Okay, RNA encodes what? Proteins. These are essential for infection and multiplication inside the host cells. Here you see, okay, about TMB, its a structural description, and about first capsid, protein subunits, followed by genetic material and its components. That is it. Make sure you are drawing an effective diagram first. Okay. Then next question from viruses is explain the structure of T even bacteriophage. Remember, the questions about TMV and bacteriophage are important for your NEET and MCET as well. Whatever concept you are learning, learn it thoroughly so that you will be ready for both subjective pattern of answering and objective pattern of answering. Look at here, ma. Explain the structure of T even bacteriophage. Look at the importance of the question. Appeared quite a times in NEET 2023, March 2020 and March 2018. Bacteriophages are viruses which will infect and multiply within the bacteria. They will infect and multiply within the bacteria. The T even phases, for example, you see T4, T2 and T5. They are mostly studied bacteriophages. Especially T4 will infect what? T4 bacteriophage will infect Escherichia coli, that is E. coli. Okay. T4 bacteriophage will infect E. coli. And here is the structure of a bacteriophage, which has 4 components in it. First is a head. Head is covering what? Genetic material inside. Genetic material is double stranded circular DNA. Double stranded circular DNA, where it has a collar below it. This collar will connect the head to the sheath. Okay, it will connect the head to the sheath, and at the base of the sheath you have a base plate. Adjoining to the base plate it has what? Tails. Adjoining to the base plate it has tails. Tails will have a long tail fibers. Okay, so it has head, genetic material, collar which is attaching sheath and head. At the base of sheath it has base plate and it has tail fibers. At the end of tail fibers you have what? The enzymes like lysozyme which will degrade the wall layer of bacteria. Next you see size of bacteria. A second. Yeah, size of bacteria, the T4 bacteriophages is approximately of 200 nanometers long. Okay, it is approximately 200 nanometers long and it has 80 to 100 nanometers wide. It is 200 nanometers long and 80 to 100 nanometers wide. What is the shape and symmetry for this? T even bacteriophages, it will have complex symmetry with binal symmetry. okay, complex symmetry with binal symmetry, which means it has both polyhedral head and a helical tail. Polyhedral head and a helical tail. Head here or capsid, the head is polyhedral or icosahedral in shape, and this is made of what? Protein subunits. This is made up of protein subunits. It encodes, this encloses what? Double stranded circular DNA as the genetic material. The head is connected to tail with a short neck or collar. Describe it in a systematic way. Next is tail region. It is cylindrical, composed of what? Contract tail sheath, surrounded by central hollow tube. The tail connects base plate to head. Base plate is hexagonal disc. Okay, this is hexagonal disc which is located at the end of the tail. This will act as an anchoring structure. This will act as a what? Anchoring structure during what? Attachment to bacterial cell wall. Fine, right? Tail fibers and pins you see here. The base plate will bear 6 long tail fibers and 6 short pins. 6 long tail fibers and 6 short pins. These fibers will help bacteriophage to recognize and attach to the host cell surface. Fine, right? Everything is clear till here. Lastly, function of tail sheath and then about genetic material. Function of tail sheath, then genetic material, then symmetry type. Function of tail sheath: during infection, tail sheath will contract. Okay, during infection, tail sheath will contract. This will drive the hollow core through the bacterial cell wall, allowing what? Allowing infection to the viral DNA into the host cell. Okay, next genetic material you see, the genetic material of T-even bacteriophages is double stranded DNA. This will carry all the necessary genes for replication and synthesis of viral components. Next is what ma? Symmetry type. The head shows what type of symmetry? Polyhedral symmetry, while the tail will show helical symmetry. Okay, tail shows helical symmetry. This together will form what? Binal symmetry. Okay, polyhedral head and helical tail will show what? Binal symmetry, which is unique to the bacteriophages. Last but not the least, which is about lytic cycle. Okay, we have 2 life cycles for viruses. One is lytic cycle, which will kill the host cell. Second is lysogenic cycle. The life cycle of virus will continue without the death of the host cells. So here you see lytic cycle. The name itself is saying lysis, which is death. Lysis, the lytic cycle is replicative process of virulent bacteriophages in which host bacterial cell is destroyed or demolished after producing new viral particles. The T-even bacteriophages, for example T4 phage, will follow this cycle. See, the steps are important over here. Always the steps are important. And what type of bacteria will follow lytic cycle beta? T4 bacteriophage mostly. Okay, attachment of the bacterial plates or tail fibers to the wall of the bacteria. And at the tail fibers, what enzyme is present? I told you, lysozyme. This lysozyme will degrade the wall layer of the bacteria. This first thing is what? Attachment. Next is what? Penetration of the double stranded DNA into the host cell, that is bacterial cytoplasm. Second is penetration. Third is biosynthesis, that the utilizing the cytoplasmic machinery or cellular machinery of the host cell, viral particles will be duplicated or synthesized. That is the third step, biosynthesis. Fourth is maturation. All the synthesized particles like the head, tail and the genetic material will come up into one single viral particles, which is maturation of viral particles. Next, last is lysis. Till biosynthesis, the process of lytic cycle and lysogenic cycle is same. From maturation, it will be different, okay, because all the cells will, all the viral particles will mature in a single cell and they will lyse the cell and they will break out. But in the lysogenic cycle, after biosynthesis, after penetration, each viral particle, the host cell itself will replicate, which will lead to the formation of two viral particles separately in two separate host cells. As such, the cycle will continue. Whereas in lytic cycle, all the viral particles will mature in a single cell, which will lead to the death of the host cell. Remember the difference, simple difference between lysogenic and lytic cycle. Here we go with attachment first, attachment or adsorption. Bacteriophage will attach to the surface of the host bacterium. Okay, it will first attach to the surface of the host bacterium, that is E. coli. This attachment will occur when tail fibers of phage will bind specifically to the receptor sites. Okay, this step will ensure what? Virus which infects the suitable host. Second is what ma? Penetration, we have learned. It will release out the double stranded DNA into the bacterial cytoplasm. Fine, right? Like how from syringe the injection is dwelled or given into the host body, the same way like a syringe. Okay, tail fibers will be adjoining to the bacterial wall and that will be given out the DNA to host cell. The protein coat will remain outside. That protein coat is addressed as what? Ghost. Protein coat is addressed as ghost. Next is biosynthesis. Once inside the viral DNA, this takes control of the host cell machinery completely. The host DNA and the protein synthesis are suppressed. Host DNA and protein synthesis will be suppressed. Next, the viral DNA directs the cell to produce new fast DNA molecules. Okay, this will produce new fast DNA molecules, proteins and enzymes. There is no complete fast particle which are formed at this stage. This phase is known as what? Eclipse period. Okay, this phase is known as eclipse period. Remember, all these terms are important. Examiner will be looking for each and every step more specifically. Please mention out at each step, every point, every detail is important. Next is what? Maturation, and last is release. See here carefully. Maturation. Maturation is nothing but assembly. All the viral particles that are synthesized inside the cell, they will unite and form into one virion or virus. Okay. New fast DNA molecules, they are packed into newly synthesized capsid heads. Tail sheets, base plates, tail fibers are assembled. They will form one complete virion. This is infective virus particles. Last is what? Release or lysis. The host bacterial cell wall is broken down by viral enzyme. This viral enzyme is called as what ma? Lysozyme. The cell will burst open, releasing out hundreds of new bacteriophages. Clear? This will be releasing out hundreds of new bacteriophages. These new virions can then infect other bacterial cells as well by repeating the cycle. Okay. This is the complete sequential step now. First follows what? Infection, penetration, first is infection, attachment or adsorption, next penetration, biosynthesis, maturation and release. Fine, right? With this, short answer type of questions from this chapter, bacteria and virus, from both the chapters are done. I would like to sum up the session by giving you one more time the details about the books, IPE books that are being published from NEET World, which are completely for free of cost, available to all the people, to all the students. What is the procedure that you can get the soft copies of these books? First you have to do is just follow us both in the Telegram and Instagram channel, where you will also get some creative shorts, reels and stories on the toughest topics you are struggling out there. And then go to Play Store, install MedWorld app, just say hi in the chat box, all the 4 PDFs will be dropped to you. Follow us on YouTube as well, you will get complete content for your MCET, NEET and IPE exams. And here To know more about the details or programs that are happening inside the institute, you can call us to the given number, or you can visit our official website, which is www.neetworld.in. This is the complete information about the books that we can give you. Okay. And finally, from my end, I can strongly suggest to all the students of 12th BIPC, 12th standard, dear students, please don't just mug up the questions of IPE. Okay, don't mug up the answers of IPE. Learn the subject or content in such a way that you will be ready for both subjective type of exam and objective type of exam as well. Practice all the set of objectives, okay, to whichever content you are learning for IPE, practice all the set of objectives, so that parallelly your MCET and NEET preparation also will move on. Okay. So, that is all from my end, Vita, today. Thank you.